So our next speaker is Jessica Morgan, who is Associate Professor of Ophthalmology and a world-class expert on adaptive optics OCT imaging. And the title of her talk is Optoretinography in Choroideremia.
Great. Thank you so much, Josh and everyone. I'm really excited to be here and talk to you about our work today. And I'll just mention, like, Eric, wow, what a really fantastic lecture to kick off this great day of scientific content. My lab also thinks very hard about clinical trials, the right people to enroll, and how to get those early markers of early biomarkers to show whether a treatment is having a physiological response, even if we have to wait those longer time periods to see changes in vision. And so we approach that from a different area, all related to imaging. So before I get into that, I want to certainly thank all of our funding sources, in particular the NIH, and also the Choroideremia Research Foundation via the $1 Million Bike Ride that has been supporting this effort. And I also want to make sure to acknowledge all the wonderful people in my lab and collaborators that make this work possible. In particular, my graduate student, Angela, is responsible for much of the data that you're going to see today, and so a special shout out to her. All right. So my lab is involved in retinal imaging, and ophthalmoscopy has been a large part of ophthalmic exams for a very long time because it allows visualization of the retina in our living patients. But many of the diseases, in particular the inherited retinal diseases that Eric so beautifully talked about earlier, affect cells or are cellular defects that cause changes at that cellular level. We can't just zoom in to a fundus photograph to see individual cells because the eye's optical aberrations cause distortions in our wavefronts that limit the resolution of our images. And so my lab uses a technique called adaptive optics, where we first measure the eye's aberrations and then correct them with a deformable mirror. And that allows us then to acquire a high-resolution image, so much so that we can now visualize individual cells in the living retina. And I don't know if I have a mouse. So when we've zoomed in here, now this image is of the very center of the fovea, where each individual bright spot is an individual cone photoreceptor in the living eye. And as we move out, in this case, to the nasal retina, you see that the cones enlarge, and then they become interspersed with the rods, which are these smaller cells in between. When we think about gene therapies are aimed to restore function to cells that are structurally present. And so I think about these images in a way where we can detect which patients have remaining cells, i.e. what are good patients that could be enrolled in our trials, that they might be amenable to a therapeutic approach. But just because we see that a cell is present in our image doesn't mean that that cell is functioning. And so my lab has also been working to develop a physiological biomarker for assessing cell function through our imaging techniques. This recently has been termed optoretinography, and the idea is that we can measure an optical signal that arises from the photoreceptors in response to a visible stimulus. So here is just an example video sequence with the adaptive optics SLO, where in the middle of the sequence, we stimulate all of the photoreceptors with a green light, the whole imaging field. And then we track the cell's intensity throughout the course of this video. And so here, two cells have been highlighted, one in the blue that starts with an initial intensity, and that green stimulus comes on, and the cell's intensity increases in response to that light. Now, interestingly, just a neighboring cell outlined in that orange also starts with an initial intensity. We stimulate that cell at the same time, it's the full field, and that cell's intensity decreases with the light. And we spent several years working with this technology and developing a way to quantify this signal. I'm going to skip over that entirely with the methods, but I'm happy to discuss it in detail after or in the Q&A. But essentially, what we're going to do now is measure the dispersion of intensities across all of the different cells in the field. And when we do that, we're then able to pull out what we call the optoretinogram, or the ORG. So this trace here is the ORG for that image of cells with the ORG on the y-axis and then time from the stimulus on that x-axis. And we then undertook a number of studies to show that this signal was indeed a biomarker or physiological marker of cone function. So the first experiment that we did was just look at a dose response. If we increased the stimulus intensity, we found that the ORG response also increased. We then varied the stimulus wavelength in order to show that the population cone optoretinogram, that action spectrum matched the human photopic luminosity function, or the spectral sensitivity of the eye. If we want to have a biomarker for function, it will need to be repeatable. And so we undertook a study to show that this ORG amplitude was repeatable both within and across sessions for our subjects. And more recently, we've now been looking at the different responses that you get at different retinal eccentricities. So studying how the ORG changes in different areas over the macula. And so all of these lead to the idea that we could use optoretinography as a marker of function. And so as we continue to develop the technique, we're also starting to translate this technology into our patient populations. And so the group that I'm going to talk to you about today are patients with choroideremia. It's an inherited retinal degeneration caused by the CHM mutations, which are located on the X chromosome. There's a prevalence of 1 in 50,000, and patients typically present with nyctalopia, constriction of the visual fields, and then finally, in later decades of life, loss of acuity and legal blindness. So previously, my lab has done many studies in choroideremia, and we have initially shown that the ORG amplitude was significantly reduced in the CHM paraphobia. And we have also studied just the structure of the photoreceptors in this group. And so there was a large study undertaken in the lab with, I think, more than 50 CHM patients, this is a rare disease. That is a high number of choroideremia subjects included. Looking at the foveal phenotypes of these patients. And so shown on the left is a normal adaptive optics fovea, whereas on the right is a patient with choroideremia. And many of these patients show what we call a salt and pepper phenotype, have these pockets of hyper or hypo-reflective photoreceptors. And so we believe that these different phenotypes show different severities or increasing severities of choroideremia. And so now we wanted to test whether our functional measures of optoretinography would also show up in these different phenotypes. So we collected the adaptive optics images across surrounding the fovea and then out along the retinal meridians, and identified a region where we were going to look at the optoretinography signal. We used a 545 nanometer stimulus, which was just shown for 0.2 seconds. So it was like a flash over the full field. All right, and then we align all of those images, identify the cones, and extract their intensity signal throughout the video sequence. Again, arriving now at this ORG signal over time. All right. We had 10 participants in this particular study. 2 were normal-sided controls, and then we had 8 CHM patients. 2 of those CHM patients were categorized as having a relatively normal foveal appearance on the AOSLO image, whereas 6 of those individuals were characterized as having the salt and pepper phenotype in the foveal region. So when we look at the results from the ORG, first we found that the two patients with our relatively normal foveal phenotype did not have a significantly different ORG in comparison to the normal-sided control participants. But you do see that the two subjects there had a rather large variability between them. And so when we looked at those images, when we look at those two, we see what appears to be that relatively normal mosaic. But if we zoom out and look at the larger scale AOSLO image, what we find is that while the fovea itself might have that relatively normal phenotype, the patient with the lower ORG was actually starting to show the salt and pepper phenotype even at locations outside of the fovea. And so we do believe that the person here represented by the bottom image is undergoing that continuum of disease severity with the advancing scale. All right, then we looked at the 6 patients with the salt and pepper phenotype and found that those individuals showed a significantly reduced ORG amplitude in comparison to the other groups. And so if I now look just at these locations, we took all of those images with that salt and pepper phenotype and separated them into the hyporeflective or hyperreflective patches. So in other words, the salt versus the pepper. And then we wanted to look at the optoretinogram response of those 2 different groups of cells within the image. So when we do that, we actually see that both of the hyper and hyporeflective cones show that reduction in ORG in comparison to the 2 other groups. And we can go and look now at the comparison between these 2 groups. And what we found was that actually with a pairwise comparison, the hyporeflective patches showed a significantly reduced optoretinogram in comparison to the hyperreflective, though the magnitude of this difference was small. So we've begun now to tease out what is the cause of this difference. And so we started by looking at the cells' reflectances over that whole video sequence, looking at the range of what a cell intensities can be for these 2 different groups. And so plotted here is a histogram just showing the maximum cone intensity minus the minimum cone intensity for each cone in those 2 groups. And what we found is that those hyperreflective cones actually on average are dimmer, have a lower range than what the hyperreflective cone would be. And if we then look at the optoretinogram amplitude as a course of throughout this histogram, we do find that the difference of these of the ORG is for the hyperreflective patches is likely caused or driven by the cones that have these higher signals. And so there's still a question remaining now about whether those cones really are functioning different than the others. And so we're thinking about ways to tease out psychophysical responses by using things like adaptive optics microperimetry, where we could direct our stimuli very, very precisely to the specific cones of interest. So with that, I'll conclude just to say that optoretinography was in the relatively normal foveal phenotype CHM patients was similar to normal-sided. The optoretinography amplitude correlated with disease severity, and those hyperreflective cone populations show a small but significant difference in comparison to the hyporeflective patches. So thank you very much for your attention.
Thanks, Jessica. Questions?
Yes, Eric.
Thanks. That's really impressive results got there. Very cool. I know you're trying to figure out what makes the cones hyper and hyperreflective, but do you have any sense of what's happening physically to change their reflectance?
Yeah, that's a great question. So an adaptive optics SLO image, in particular the ones that I showed here, which are confocal. So we have a confocal pinhole in front of our detector that's placed conjugate with the retina. the AOSLO, that confocality does give us some actually axial sectioning, but not nearly to the extent of an OCT. So when we look at an AOSLO image, such as the one I showed, we believe that the cone inner segment, outer segment junction, or ellipsoid zone, and then the cone outer segment tip, or that interdigitation zone. Those are the two layers of the OCT that we believe are kind of combined together and appearing in our AOSLO image. And in choroideremia, we do know that that interdigitation zone, or the cone outer segments, are disrupted. And so I think that certainly we are seeing some of the abnormalities that we see in the AOSLO images are manifested in those retinal layers.
But that also implies that that something about that interface is changing following exposure to light, right?
Yes, so we believe the mechanism for the ORG is actually caused by when the light hits the photoreceptor. Let's take a normal-sided control for now. When light hits the photoreceptor, the distance between those two layers changes, or I say the outer segment path length between those two layers is altered. And that can cause interference between light reflected from those two different surfaces. That is then what leads to the effect that some cones can get bright, some cones can get dark, still with the same stimulus. And so certainly when you now expand then to the disease where you have altered structure also, that mechanism can also be altered as well.
Thanks.
Absolutely.
Jessica, great talk,
Thanks.
as always. Thank you. Since you didn't have time to go into the mechanism of what you're measuring, can you clarify that the population ORG, is that the average change in signal up and down across the area or per cell?
Great question. Okay, so what I showed today was the population ORG, meaning we're taking the signals from individual cones but aggregating them together. And that's why the population ORG matches the spectral sensitivity of the eye in total. Now, if we did it at a single cell level, which we can do, what you find is that each cell you can assign an optoretinogram amplitude or trace to, and that we could use this technology to cone type. So we could say which cell is an LM or S cone, and that has been demonstrated using optoretinography, but with a complementary technique of adaptive optics OCT rather than SLO. Yeah.
That sounds exciting.
It is very much so.
Tomas.
Beautiful work. As you know, I'm mesmerized by your presentations always, but I had a question. Given that these changes are so slow over time, compared to the changes that mediate vision, which are usually in milliseconds, is there a difference in the kinetics between not only in the amplitude of the ORG, but in the kinetics between normal or at least apparently normal CHM population of cells and those who are clearly abnormal?
Right, yeah.
To a normal.
Great, yes. So are there, what I would say with the ORG is that we're still in relatively early developmental time, and the field in general, in terms of what measure we're going to extract from these traces. So comparable to an ERG where you have amplitudes, but you also have your implicit times, the ORG would have those same potential extraction metrics that we could do. And so far, I would say my team has not investigated the time aspect of those signals, in part because we're still trying to work out the best stimulus and protocol for how we would, undertake these. But anyway, the answer to your question is yes, we would expect that those metrics could come from the signal, and further that patients would look different.
So Eric was talking about looking for early biomarkers of response to therapy using molecules levels. And it seems like the ORG could also be a biomarker for early response to therapy. What do you think are the prospects for that?
Yeah, great. So absolutely, I think that it's possible. Similarly to Eric, I would not suggest this as a primary outcome, but as a secondary outcome that can justify the longer clinical trials, absolutely. The idea that you can use adaptive optics imaging, first of all, to identify patients most amenable to therapy in the pre-enrollment stage has really high potential and has not to date been used. But for these individuals that have cone structure in their fovea, the images that we show could give you a predictive tool for even how much acuity a person could gain based on how many cones they have, assuming normal wiring to the brain, right? So selecting patients for trials, absolutely. And then being able to show whether the cells have a physiological response to our treatment in order to justify these longer times. So whether that's a change in the optoretiноgram or even just a change in the cell structure visible itself. And we do have hints of that in some of the trials, even that Eric put on the screen, that ultimately did not meet their primary endpoint.
Great, thanks, Jessica.
Thank you all. Thanks so much.